So we're going to be starting this topic of cell division. Cells go through a cycle in their life. Sometimes, some point in their life, they're going through this quite often, and then they finally get to a point where they kind of stop in one of those, kind of like what we would call the interface part, because there's no longer a need for them to divide and multiply. So the cell cycle is broken up into phases that we give different names. The first phase is called interphase. It's the period of time between a cell finishing dividing and the next time it's going to divide. So that's that period of time in between. It's not a time when the cell is just resting. The cell actually has a lot of activity going on during that time. It's going to be growing, as it says right here. It's usually growing in size, and we said that it increases in volume, and it also increases in surface area. But what limits the size that a cell can get? That was one of our questions on the test. Yes?
Surface area.
Okay, the surface area. When the surface area can no longer supply everything that the cells need, then the cell's at the point, probably like right over here, where it needs to go into mitosis. Up to that point, it's just manufacturing all the things it needs to grow. Then, when it's ready to go through a division process, it goes into what we call mitosis. And what's the primary thing that's happening in mitosis is the DNA that's in the nucleus is going to get duplicated so that we can divide it up into the two new cells that are going to form as a result of mitosis. So the nuclear material gets divided between these two new cells that are forming. And then once that has happened, we need to divide the cell, like kind of pinch it off or divide it off or wall it off, whatever you want to call it, between the two halves so that we have two distinctly new cells. So let's talk about this time in between divisions that we call interphase. The interphase is actually broken up into three phases of its own. We have the G1 phase. This is where the cell, as it says up there, is doing a lot of growing. It's increasing its cytoplasm by also increasing the number of organelle that it has in there, or the size of the organelle. Whatever the cell needs to have more of, it's going to make more of, and it's going to get the information from the nucleus to make that. The S phase, which follows that, is a phase where the DNA is replicated. So in other words, the cell is grown to the point where it no longer can grow in size. Then that will cause the cell to duplicate its nuclear material. In other words, the number of chromosomes will need to be doubled, because we're going to give one set to one cell, or one daughter cell, and the other set to the other daughter cell. So we need to double everything so each cell can have a complete set of information. Then the final one is where the cell is kind of setting up the stage for cell division. So we say preparation for mitosis, where the nuclear material is going to be separated between like the two parts of the cell, the two halves of the cell, opposite ends of the cell. Okay, so whatever the cell that we start with, we often refer to that cell as the mother cell. So wherever you are in this, in this going through the cycle, if we look at that cell at that point, we're going to call it the mother cell. And then the cells that form from that we're going to call daughter cells. Now, each of these daughter cells will then, could become another mother cell, and then will divide into daughter cells eventually. Okay, so mitosis, we said, is the point where the nuclear material, now the chromosomes in the nucleus of the cell are going to be divided. Okay? We're actually going to divide it up. And there's a process that will go on. And one thing we need to remember is that the nucleus at this point has a identical copy of all its information. So basically it has two blueprints. Instead of having one blueprint, it now has two, because we're going to divide the mother cell into two daughter cells, and each one needs its own blueprint of everything that's necessary for that cell's growth and survival. So let's now look at prophase and divide it into phases, okay? Or not phases, let's mitosis and divide it into phases. The first part of mitosis is called prophase. Okay, what's happening in prophase? Well, the chromatids that we've talked about before will actually become shorter and thicker. Okay, the chromatin material up to this point was kind of, you couldn't see distinct chromosomes, but at this point they're starting to come together, thicken up, coil up, and so you're going to actually start to see chromosomes inside the nucleus of the cell. Another thing that's happening is we need to get rid of the nuclear membrane, the thing that holds all the nuclear material in one spot. We need to get rid of it because it's going to interfere with the cell's ability to separate it into two groups. So we need to get rid of the nuclear material. So that's another thing that happens in prophase. The centrosomes, remember that was a structure that was outside the nucleus, out in the cytoplasm, is going to migrate to kind of like split and migrate to opposite sides of of where the nucleus is, has been, or is, I guess it still has, just nuclear material is gone, membrane is gone, but it will to the opposite ends, and then between these two centrosomes, or centrioles, it's going to grow sort of like some microtubules, like fibers, and we call it a mitotic spindle at that point. To these little fibers, the chromosomes at their centromere are going to be attached at that spot. So like get glued on to there at that spot. So when you see in a cell, the chromosomes have already thickened up and you're already starting to see them inside where the nucleus was. When they start to line up at the center, sort of like in American football, we have everybody lines up at the line of scrimmage. Now, here's where the difference is going to be, is in American football, they're trying to run through each other, right? Well, in mitotic division, they run back to their own, where they, I don't know if you can call it goal line, because you're trying to get to this goal line, but they actually like run away from each other. So they're lined up first in the very middle, with the centromeres holding the chromosomes attached to the spindle at the very center. Now, the part that makes it a little bit different, you have to remember that this is in three-dimensional space. So instead of the cell being flat, it's has size, and it has a plane going through it. You remember what plane is from like geometry? A plane going through it, and all these chromosomes are lined up along this plane. So when you look at it on the edge, it looks like they're all in a line, but if you looked at it from a different perspective, maybe the end, they would look like they were lined up in a circle. But they're, so it would be like a line of scrimmage, but in a circle instead of a straight line. Then the next thing that's going to happen after they have lined up in the middle is the chromatids, these two of them, are going to separate from each other. Okay, so we have double the material, and now we need to start reducing the amount of material that's going to go to each, or divide it up, would be a better way of saying it. Divide up the material so that this half of the cell will get half of the instructions, or remember we doubled it. So it'll still be a full set, but it'll be half of the chromosomes that exist at this point, and these will get the other half. So these centromeres, where the chromosomes are kind of like attached, will pull these daughter chromosomes to opposite ends. So here you can kind of see that they're moving away from each other toward opposite ends of the cell. This is what they would look like if they were lined up in the middle. You can these little blue lines that are in this picture are like the spindles. They're clear, they've been stained, but they're going to be pulling these chromosomes to opposite ends. The daughter chromosomes will actually move to the end of the spindle. The spindle is kind of like this. So they'll move to the opposite ends of the spindle, and then so they've already arrived then when that happens. So we need to keep them separate from the rest of the cytoplasm now. So the nuclear membrane is going to reform around that group. That will become that new cell. That part will become a new cell eventually, and this part will become a new cell. And then these daughter chromosomes, they were all coiled up and bunched to look like a chromosome. They'll now start to unwind themselves because they don't need to be tightly bound up like that. I would think, I'm not sure, but I would think that would interfere with the ability for transcription if they were all bunched up, so they need to unwind to allow transcription to take place, which is going to happen because we want this cell to grow. So once they have two nuclei formed on opposite ends of the cell, then it's time to divide the cytoplasm in half. It isn't exactly half all the time. We'll actually talk about a kind of cell division where it's not exactly half. I think your book actually shows some pictures where one will actually get all the cytoplasm and the rest of them get very little. Well, these will typically die because they don't have enough cytoplasm to exist. So we put sort of like all our energies or all our eggs in one basket, and these others will die. But there's a reason for that. It typically happens in a type of cell division that we call meiosis when you're forming eggs. So this is what females do. Males, on the other hand, we want everyone to get the equal amount because we want them all to survive. We need huge bunches of sperm to make sure that the one egg gets fertilized. So in animal cells, they will pinch off in the middle. We call it, they cleave, like a meat cleaver. You use it to cut meat, like that. So think of it kind of pinching off in the middle of the animal cell. And so that now we'll actually have two separate cells. The cell we started with was the mother cell, and we'll end up with two daughter cells. If this is a plant cell, instead of cleaving and having this, what we call a cleavage furrow, this indentation around it, it'll actually grow a wall in between. And now we'll have two separate cells, and a wall divides the two into two separate cells. So let's talk about a little bit how mitosis can vary between plants and animals, between multicellular organisms and single-cell organisms. Okay? So the cytokinesis, the part of dividing we saw was different. Animals kind of pinch off, plants create a wall in between. Animals have a centriole, plants don't. They accomplish that without using a centriole. In unicellular, which means what? What does unicellular mean? One cell, right? One cell. Sometimes they don't go through cytokinesis. In other words, sometimes they don't actually divide their cell into separate cytoplasms. So that means you're going to end up with a cell that will have more than one nucleus. But because it has more than one nucleus, it can actually become a little bit larger because of that. So you can have this kind of like, it's more like a blob that, the cell that's large, that actually is, but different nuclei nearby or controlling that area instead. So it actually doesn't divide into individual cells. But that's only some one-cell organisms do that. Not all of them, some of them do. Now, all cells take a certain amount of time to go through that cell cycle, and it varies. Some of them go through it very fast, and they grow really quickly, and others do it more slowly. Like, for instance, some aggressive forms of cancer they will grow and not grow to, to a full size before they'll go through the division process. And so they'll do it really fast, and so the cells will multiply quickly, like in an aggressive form of cancer. Your cells don't usually multiply that fast. That's the reason why it's taken how many years to get to you this size, or your mom carried you in her womb for 9 months to make you big enough to be born, anywhere from 5-6 pounds up to 8-9 pounds, something like that. The length of time it spends in interphase kind of varies from one kind of organism to another, or one kind of cell to another kind of cell. And then the number of divisions that they'll go through. Some of them will divide, constantly. Like you have certain parts of your body where the division is just happening on a regular basis. Others where, once it gets to a certain point, it stops. It doesn't reproduce itself. Like your nerve cells, your neurons in most of your body don't reproduce themselves in the spine, the brain. So once those cells die, they're not going to be replaced. So whatever you do to kill some of your brain cells just aren't replaced. So that's the reason why we have things like for certain sports. Like rugby, I think, is one of those where you can only have about so many concussions, and then they won't allow you to play anymore. Because we know you don't replace those brain cells. It's turned out more recently that we've discovered that people who play soccer and head the ball also experience a bit of brain damage every time they head the. So, it's maybe not as bad as a brain concussion, but it is a minor one all the time. Now, multicellular organisms, what do they use mitosis for? And we are a multicellular organism. What do we use mitosis for? Well, one of them is for growth. This is how you got to be as big as you are right now. It's also for repair. So if you injure yourself, fall down and skin your knees, your arms, your elbows, or something like that, your body uses this type of cell division to replace the cells that were damaged. Also, to replace worn-out cells. Like, for instance, your red blood cells only last about so long, they become worn out, and they have to be replaced. So your body will use mitosis to replace those red blood cells so that you constantly have enough to carry oxygen to all the various parts of your body. Now, in some organisms, it's also a way of reproducing themselves. For instance, we can take a starfish and chop it into pieces. And as long as a piece of that has what we call the central canal, when we study about starfish, you'll learn about this in detail. But if as long as it has a piece of that, it will grow all the parts of the starfish that it's missing. So chopping a starfish up into pieces to kill it and throwing it back in the water, it doesn't kill it. It just makes you have more starfish. And starfish, their favorite food is like clams and oysters and stuff like that. So if you were growing clams and oysters for food, you definitely want to get rid of the starfish, but that's not the way to chop them in little pieces. You don't get rid of them by doing that. So it can be used as a form of reproduction. In plants, have you ever seen someone do that? Maybe you've done that yourself, is taking a cutting from a plant and maybe put it in water or in moist sand or something like that, and it will grow the roots. Remember, every cell has all the instructions on how to grow the pieces that are missing, as long as those genes can get turned on. So typically in those plants that we can do that, losing the roots will turn on the genes for growing roots. And then when it finally grows to a certain size, we can transplant it and grow into another plant. Now, this is the kind of thing that you have to do with plants that don't reproduce themselves but ordinarily do, like seedless oranges. How do you get more oranges if it's seedless? Well, we have to use this process to reproduce those in a way, used by making a cutting and growing those and then planting those, because they can't reproduce themselves. We also do that if you have a garden and you grow potatoes. If you have a garden and you grow potatoes, what do you typically do when we say plant potatoes? What do we do?
Normally bury part of the potato.
Do you bury the whole potato?
Well, normally like where it makes an eye.
Yeah, you cut it into pieces, and as long as a piece of it has an eye, we call it, right, in the potato, then it will grow into, because the eye is actually a little miniature plant right there, and then the fleshy material of the potato will be the early food that it needs to grow to the point where it has enough roots that it can support itself. But if you think about it, if this is mitosis, cutting a potato into pieces, planting them, I will now have plants growing that are all identical to each other. They all have the same genetic code. So every plant is exactly alike. Genetically. Doesn't mean they have to look exactly alike, because remember, environment is part of that. But they are genetically alike, which can be a good thing. If it possesses the characteristics that I like, for that kind of potato, then all the potatoes are going to be that kind. I like the flavor, I like the, how tender the flesh is or something like that. That'd be a good thing. But the downside is, what if that particular variety is susceptible to this disease that suddenly moves in? Then they're all affected, right? None of them they're all susceptible. So having a variety sometimes is a good thing, as far as preventing disease from wiping out everything. So typically, like in most people's lawns, we put in many different varieties, because if one of them is susceptible to disease, maybe the rest of them aren't, and I'll still have a lawn. So asexual reproduction, this is the process by which organisms will reproduce themselves without having a sperm and an egg. So that's what we mean asexual, no sexual part to this. So they're going to use that mitotic cell division for that. The thing is, we're not going to have any genetic variety. They're all alike. And here were the things that we've kind of talked about. Fragmentation is like chopping a starfish up into pieces. Budding would be cutting a piece of a plant off and maybe grafting it in or doing something like this. This is what, for instance, like mushrooms would do. Mushroom grows up, produces spores, the spores spread, and every mushroom that comes from that spore is going to be like it. It's going to be genetic identical to the others. Okay, so I'm going to show you here it's a teacher that's kind of doing a simulation would be better, not an animation, but a simulation of mitosis. And then he's also going to show you meiosis. I want you to look at what's the difference between these two as he does that. So we'll see if we can get this going. Had it working earlier, so we hope it will work again. Oops, not seeing it.
Okay, today I'm going to model mitosis, and we're going to do it with a simple organism.
We'll have to do what we normally do. Hopefully this will be. Can you see it? Yep.
In this organism, it's going to have, we'll make it 4 chromosomes, and I'll add those other 2 in just a second. And so our diploid cell is going to have a diploid number of 4. What does diploid mean, haploid mean? Well, in this case, diploid means that you have a chromosome from your father, that you get from your father, and a chromosome that you get from your mother. Now, we would call this chromosome 1, and that's because it's the longest one. If we have 2 other chromosomes, this would be chromosome 1, and this would be chromosome 2. Now, in us, we have 22 what are called autosomes, and then we have sex chromosomes. But the way chromosomes are named are just their number. 1 is the longest, 2 is the next longest, 3 is the next longest. So a few other things you should know about a chromosome. This would be the centromere, the center where they're connected. And then each of these beads represents a gene. Now here we just have a few genes, but in a human cell, we're going to have thousands of genes on each of the chromosomes. And so this is a very simplified model. You can imagine if this is a human chromosome, my whole board would be filled with these chromosomes. And so this is a very simple model. So this one right here from dad and this one from mom are what are called homologous chromosomes. What that means is that they have the same length, but they don't necessarily do the same thing. What does that mean? Chromosome 1 from dad and 1 from mom are going to be exactly the same length, centromeres locate in the same thing, and the genes will be in the same spot. So for example, if this is the gene for blue eyes, this would be where the other gene for blue eyes is. So you may be asking yourself, well, how are they different then if everything seems to be the same? Well, this right here could be a recessive gene for blue eyes, and this could be a dominant gene. It doesn't give you blue eyes. And so those alleles are going to be found on either side. Or this right here could be the gene for hitchhiker's thumb, makes your thumb bend backwards like this. And over here, so hitchhiker's thumb here, this could be one for a straight thumb, so you don't have a hitchhiker's thumb. And so the chromosome you get from mom, dad, and the chromosome you get from mom are homologous, and they never ever meet, except in meiosis, which we'll get to in just a second. Okay, so what's our goal in mitosis? The goal in mitosis is to make an exact copy of the nucleus. And so this circle right here, bounded by this, is going to be the nuclei, and these are going to be the chromosomes inside it. Now, what does a cell do? As before it divides, a cell will get bigger, and then it will copy its DNA. So let me model how that works. When it copies the DNA, this chromosome will have an exact copy of it made. This occurs during the S phase. This chromosome will have an exact copy of itself. This one will have an exact copy of itself, and then this one will make an exact copy of itself. So maybe this is what you remember chromosomes looking like. And so after the S phase, you'll eventually have a cell or a chromosome that's made an exact duplicate of itself. And so if this is a gene here, there's an exact copy on this side. In other words, this side and this side are exactly the same. In fact, those are called sister chromatids at this point. And so this would be at the end of the S phase. What happens after the S phase? It goes into another growth phase called G2 phase. So G2 phase, all of that is part of interphase. Now, would it look like this during interphase? You wouldn't see the chromosomes during interphase. All of that DNA would be loose within the cell, doing its job, the job that it normally does. And so you're really not going to see chromosomes look like this until we get to prophase. Okay, what happens during prophase? All that DNA is going to coalesce and we'll actually be able to see it. Okay, what happens next? So that would be prophase. The next thing is going to be metaphase. What happens in metaphase is that little spindle fibers will attach to these, and they will line up and meet in the middle. And so they're going to line up like this. This chromosome will line up like that. This one will line up like that. And this one will line up like that. And so the reason they line up like that is that the spindle fibers attached to them,
So that's that mitotic
they're going to
spindle.
go from here to here, from here to here. And then we're going to have attachments over on the other side as well. Now these spindles are going to go outside of the nuclei, which I would draw, but I can't quite fit that on my screen right here. And so there's going to be a tug on either side by the spindles, and that lines these all up in this perfect, what is called the metaphase plate that goes right down the middle. So this would be metaphase. What happens to next? The next is going to be anaphase. So in anaphase, what happens is these are going to be pulled to the side as those spindles shorten. They'll be pulled like this. These will be pulled. This all happens at the same time. These will be pulled like this as well. So they're going to move to the sides, and then the spindle fibers are going to start to disappear. And then we're going to have two brand new nuclei. So this would be one nuclei, and this would be another nuclei. Now, if you look at the chromosomes in each of these nuclei, we've got one like that, we've got one like that, we've got one like that, we've got one like that. They're exactly the same. In other words, we have two brand new nuclei, and each of them have the same duplicate DNA in each one. And that's the goal of mitosis. The goal of mitosis is to make two exact copies of the cell. Okay, so I'm going to take a minute and clean this up for just a second, and then I'm going to show you how that differs from meiosis. So in mitosis, we're making two nuclei, and those nuclei are going to be exactly the same. But now let me show you what we do in meiosis. In meiosis, our goal is not to make duplicate cells, but to actually make different cells. We make genetically different cells. And so how does it start? Well, it starts the same way. The cell's going to make a copy of itself, and so it's going to go through a growth phase, so the cell will get larger. It's then going to duplicate its DNA, so it's going to duplicate its DNA like that, and like that, and like that, and like that. So this is going to be at the end of interphase. So the cell has grown, it's copied its DNA, and then it started to grow again. But now we're going to have something different happen. And so in meiosis, we have two divisions. And so during the first division, what will happen is the homologs will come together. And this forms something called a tetrad. And so we'll have this homolog together and this homolog together. In other words, the chromosome that you get from your dad and the chromosome that you get from your mom will actually come together, and they'll wrap around each other like this. They line up along the middle, so this would be in metaphase 1. And they're going to, this actually happens a little before this during prophase, but what will happen is that they're going to wrap around each other so closely that portions of one will switch with portions of another. And portions of this one will switch with portions of another one. And so during prophase 1, they're going to switch bits of them. Or this one right here, it might pop off here, this little gene, and it might switch with another gene over here. So it'll switch like that. And what that gives us is variability. And so this would be during prophase 1, we cross over, and so we switch some of the chromosomes. Another important thing happens at the next step, which is metaphase 1. This might line up like this, but it also might line up like this. And this might line up like this, but it also might line up like this. And this is called independent assortment, as I start to lose one of my chromosomes here. In other words, depending on which side they line up on, they're going to be pulled in a different direction. Now, why is this important? Well, let's say this right here is the gene for Huntington's disease. Huntington's disease means you're going to die when you get to be about 40 years old. And if it lines up like this and goes to a sperm or an egg over here, then you're going to get Huntington's. But if it goes over here, you're not. And so this, when you draw a Punnett square, is where that actual genetic 50-50 split takes place. That's called independent assortment. Okay, so now what happens next? We're going to attach a spindle on each of these. So there's going to be a spindle here, and there's going to be a spindle here. And that spindle is going to, during anaphase 1, it's going to pull them apart. So these are going to pull apart, and these are going to pull apart to the side. Now what happens next is we form two new nuclei. So I'll do that like this. So we form two new nuclei. And now the next thing we go through is we don't go through another interphase. And so there's no interphase, but what happens next is it will actually